it happened with the unexpected identification of the first human calmodulin mutation back in 2012, work done by my PI Michael and his wife Mette. And since then, I've been equally fascinated and frustrated with the complexity of calcium and calmodulin signaling. So today, almost a decade later, we know about 28 different mutations in calmodulin, and these can occur in any of the 3 genes that encode an identical calmodulin protein. And curiously, all patients that carry these mutations present with life-threatening cardiac arrhythmias, including long QT syndrome, catecholaminergic polymorphic ventricular tachycardia, a mix of these 2 phenotypes, and in a few cases, idiopathic ventricular fibrillation. So the clinics have now recognized the diseases related to mutations in any of the 3 calmodulin encoding genes, and these are commonly referred to as calmodulinopathies. And common features involve early onset or very early onset, as well as a high risk of cardiac arrest or sudden cardiac death without prior symptoms. So with this talk today, I would like to highlight what we have come to know about the molecular predictors of these calmodulinopathies. And zooming in on the known mutations from a structural perspective reveals that the vast majority of mutations reside in the ESHAN3 or 4 in the C-terminal domain of calmodulin, whereas the N-terminal domain of calmodulin seems to be rather mutation intolerant by harboring only 2 of all the mutations. Also highlighted here in red, most of the mutations occur in calcium coordinating residues and, unsurprisingly, reduce the calcium affinity of the C-terminal domain of calmodulin. And because calcium affinity is a fairly easily measured parameter, it would be convenient if we could use this as a predictor for which phenotype a specific calmodulin mutation will result in. So here I have plotted the QT interval, which largely separates out the phenotype against the reduction in calcium affinity of the calmodulin C domain. And as you can maybe appreciate, there's a fairly good correlation whereby the reduction in calcium affinity is able to predict the phenotype. However, there are also several deviations that proves this model to be a bit too simple. And that is maybe not that surprising, given the fact that calmodulin regulates a lot of different target proteins in the heart. And it may be that these target interactions hold the key to better explaining why some patients present with the long QT syndrome and others present with CPVT or a mix of these. So out of all these interactions, the 2 primary cardiac calcium channels, the voltage-gated calcium channel CAR1-2 and the ryanodine receptor 2, have been investigated the most due to their essential role in mediating the calcium-induced calcium release during excitation-contraction coupling. And importantly, both these channels are regulated by calmodulin, who aids channel closure by a negative feedback mechanism. These channels have also been the primary focus because of intrinsic mutations in the CAR1-2 channel are associated with the long QT syndrome, whereas intrinsic mutations in the ryanodine receptor are associated with CPVT, and these are, as I said, the primary phenotypes of calmodulinopathies. So we have implemented an assay that measures the calmodulin binding affinity of the canonical binding domains from these 2 calcium channels. That is the IQ domain in relation to the CAR1-2 channel and the calmodulin binding domain 2 in relation to the ryanodine receptor 2. And in this assay, we measure the calmodulin affinity of these 2 binding domains at a broad range of calcium concentrations. And that yields this calcium-dependent binding affinity curve, where you can see there is a 1,000-fold increase in calcium affinity with, or in calmodulin affinity with calcium concentration. And the upper plateau here in the curve represents apocalmodulin docking to the binding domain, and this may or may not be physiologically relevant. And the drop in the curve here represents the calcium sensing of calmodulin, and the lower plateau represents the calcium-bound complex. And with this assay, we're able to determine the effect of the arrhythmogenic calmodulin mutations. And in relation to the IQ domain from COW12, you can see here that the long QTS-associated mutations cause a relatively large right shift in this calcium-dependent affinity curve compared to CPVT-associated mutations that cause either no or a relatively small right shift in this calcium-dependent affinity curve. And the right shift in the curve here indicates that the calcium sensing of calmodulin is impaired when it's complex to the IQ domain from the COW12 channel. And this would indicate or suggest that the long QT mechanism is through the dysregulation of the COW12 channel. And this is verified in functional assays where the calmodulin-mediated calcium-dependent inactivation of COW12 is measured using electrophysiology. And these data show that CPVT-associated mutations cause either no change in the CDI or a reduction in CDI of the COW12 channel, whereas mutations associated with severe long QT syndrome, they completely abolish the CDI. And this serves to kind of underline that the mechanism of the long QT patient seems to be through the dysregulation of the COW12 channel. And I know this is a lot of data and a bit busy, but what I want you to notice is that in much the same way as with the IQ domain from the COW12 channel, mutations in calmodulin impair the calcium sensing of calmodulin when it's complex to the canonical binding domain from the ryanodine receptor. And extrapolating these binding data to intact channels would predict a channel that releases too much calcium before calmodulin monitors the change in calcium concentration and aids channel closure. And this is exactly what we see in functional assays, where we measure the sensitivity of the ryanodine receptor to changes in ER luminal calcium. And here, the blue staple line represents the calcium release in the presence of the calmodulin wild type. And in these experiments, the results show that the ryanodine receptor channels close at a lower ER calcium level it should have been, and in that way release more calcium per opening event. And as you can also appreciate, this occurs for all the mutations, and show that all of the ones that we've tested here dysregulate the ryanodine receptor's ability to sense changes in luminal calcium. So this gives now an explanation for the CPVT causing mutations that the COW12 channel did not. But it also indicates to us that dysregulation of the ryanodine receptor seems to be a common component regardless of the phenotype in calmodulinopathies. So to sum up what I just told you here, fairly few data needs to be collected to predict the phenotype of a specific calmodulin mutation. And here the calcium affinity, or the effect on calcium affinity of calmodulin, is very important, and is a really good predictor of the phenotype, in that a strong effect on calcium affinity gives you a higher risk of presenting with long QT syndrome, and that seems to be, and the mechanism seems to be through the COW12 channel, the dysregulation of the COW12 channel. On the other end, if you have a mild effect on calcium affinity of calmodulin, the predominant phenotype seems to be CPVT, and the mechanism for CPVT seems to be through the dysregulation of the ryanodine receptor too. And in a bit of a funny way, in that if you don't present with a strong long QT phenotype, it seems that you will get CPVT. So in a way, the CPVT phenotype is unmasked by the lack of an effect on COW12 that would lead to long QT syndrome. Also, when looking at a mutation that have an intermediate effect on the calcium affinity of calmodulin, it's a bit more difficult to say which phenotype the patient will present with, and it may be a mix, as we have seen with several mutations so far. And so finally, I would like to address one of the paradoxes related to these calmodulopathies. So we now know, as I've told you, that mutations in calmodulin, they cause life-threatening cardiac arrhythmias. But at the same time, we know that calcium signaling and calmodulin decoding of these signals are required in many physiological processes that take place on vastly different time scales and with different interaction partners and interaction affinities. And this, together with the peculiar features of calmodulin, modulating more than 300 tag proteins in virtually all of our cells, together with being expressed by 3 independent genes that encode the exact same protein, which in turn does not show variations between vertebrates. That kind of indicates to us that the vertebrate world have evolved around a conserved calmodulin calcium decoding system. And to me, that raises the peculiar question, why and if calmodulin mutations only manifest as a cardiac phenotype. And it also raises the question if these calmodulin mutations have been investigated enough to say that the phenotype is limited to a cardiac one, or if there may be other phenotypes that are either camouflaged by the severity of the cardiac arrhythmias, or that may present later in life. I already told you that these calmodulin, or these cardiac arrhythmias, are very early onset in certain cases. Either it sometimes is seen even before birth or just after birth, or at least in childhood. So there may be some phenotypes that we haven't seen yet because the patients, at least many of them, are not very old yet. So I at least think that with the large sequencing efforts that are going on right now, that we will in the future be able to identify more calmodulin mutations, and these may aid in describing the full phenotypic spectrum of these calmodulopathies. And I think that's very exciting, and I also think that this paradox is rather perplexing. And with that, I would just like to acknowledge my group at Aalborg University and our collaborators at Aarhus University, Medan Anas, and Wayne Chen and Philip Van Petegem from Canada, and the late David Yu and Ivy Dick from the US. And thank you for listening to my talk.
Thanks a lot, Malene, for your talk. And we've got one question in the chat box from Rajesh. Does a changed C-lobe affinity of calcium to calmodulin also change the affinity for the N-lobe?
Can you say that again, Katja? We're just a little bit, you're falling out a bit.
Does a changed C-lobe affinity of calcium to calmodulin also change the affinity for the N-lobe?
Usually it doesn't. So not all of the mutations have been investigated in terms of affinity of the 2 lobes. For most of the C domain mutations, the N domain does not show any difference in affinity. There's a very, very small change in affinity. It's less than a factor of 2 for the mutation that actually reside in the N-terminal domain of calmodulin. We do not know, so as I showed you, there are only 2 identified mutations in the N domain of calmodulin, and we do not know yet what the effect on calcium affinity is of the other one. Yeah, so normally a C-lobe mutation does not change the affinity of the N domain, if that's the question.
Okay, so there's a question from Kevin Foskett. With 3 genes, isn't it surprising that mutations in a single gene have phenotypic consequences? And a follow-on question, do genetic and epidemiology studies suggest that patients with the calmodulin mutations are protected from or more disposed to other diseases?
Yeah, so great questions. And yes, it is very surprising that 1 in 6 alleles I mean, a mutation in an ocean of wild type, right? You would predict does have this dominant effect and even have phenotypic consequences. And I think that is also reflected in how difficult it was to actually publish this first calmodulin mutation. Like, the field just wouldn't believe that a mutation in calmodulin was something that you could survive with. Yeah, so yeah, it is peculiar. But that's also what makes it interesting, I guess. So the other question was regarding if you are protected from or more exposed to other diseases. I have no idea about that. Yeah, I also think there are very few mutations identified right now. It's very rare to have these mutations, and maybe it will be something that are being more looked into later on as we find more and more mutations.
Related to that, Colin Taylor asked, does it matter which of the 3 genes are mutated?
So it does not seem to matter in the sense, it might matter regarding the phenotype, but it doesn't matter which gene is mutated, you will present with a phenotype. But I mean, yeah, you only find what you look for, right? So maybe that's why we've only found mutations in calmodulin in cardiac patients, because now cardiac arrhythmia patients are being screened for mutation in calmodulin. But we have found, or researchers have found, mutations in each of the 3 calmodulin genes, and also the same mutation occurs in all 3 genes actually, and cause the same phenotype. So there are many interesting questions you can ask in this puzzle.
Okay, I think we'll just do one more question here, and maybe you can answer some others online.
Sure.
Lou asked whether the effects you've observed are also conserved to sodium channels.
Yeah, that's a good question. The sodium channels have the cardiac sodium channel, NaV1.5, have been investigated to some extent, but I think due to the results or the lack thereof, it didn't seem to be a very interesting target anymore. I think both the CPVT and long QT associated mutations have been tested in terms of effects on the sodium channel, and it was just one of the mutations, a very severely long QT associated one, that showed an increase in the persistent sodium currents, and you also needed to express a fetal variant of the sodium channel to get this effect. And there was another mutation also investigated, but the effect of overexpressing this one, it went away when you expressed it together with the wild type, as you would expect under physiological conditions due to the 6 alleles.
Okay, thanks for your talk and an interesting discussion. Just look at the chat box. In the interest